Buenas tardes. Bueno, muchas gracias por venir, bienvenidos a esta presentación y es un gran honor para mí presentar al profesor Samulski, a Jude Samulski. Y bueno, es difícil hacer una justicia a la carrera de Jude, más de 40 años investigando, pero voy a hacer una pequeña reflexión porque desde el punto de vista científico, una persona con 300 patentes, más de 350 publicaciones, básicamente la primera persona que clona un AV para uso clínico, la primera persona que clona un quimérico para clínico y una persona todos los productos que hay ahora en el mercado en terapia génica tienen alguno de sus patentes, alguno de sus descubrimientos. Entonces, una persona absolutamente clave que se le puede considerar el pionero de la terapia génica. Hay otros componentes también, como son el componente empresarial, donde Jude también es excelente. Él monta en 2000 Asbio, y Asbio se ha convertido en una historia de éxito, una empresa que realmente ha revolucionado el mundo de la terapia génica y que al mismo tiempo, bueno, pues hace un par de años fue adquirida por Bayer en la transacción privada más grande de los últimos 12 años en el mundo. Sin embargo, esa no es la faceta más importante de Jude y ahora veréis un poco en su presentación. La faceta que creo que más importante es el cuidado, la dedicación que tiene a los pacientes y a buscar cura para las enfermedades. Aparte de toda esa labor científica, de toda esa labor empresarial, él es el Chief Scientific Officer de la Fundación Columbus y está también empeñado en esa búsqueda de soluciones, sobre todo para aquellos niños, para aquellas enfermedades en las que no hay curas. La Fundación Columbus se dedica a eso. Tenemos una fundación en España, en Estados Unidos, y nuestro objetivo es llegar a buscar la cura para 10 enfermedades ultra raras en los próximos 10 años. Estamos trabajando con ADC, estamos trabajando con SGP50, otras enfermedades súper graves, y en las que el problema es que no hay suficiente retorno económico en los desarrollos como para justificar la inversión. Entonces nosotros desde la Fundación y Jude en particular y todo el equipo de Asbio y Viralgen ponen el empeño en que eso suceda. Y bueno, sé que muchos de vosotros apoyáis a la Fundación y conectáis con la Fundación, pero vamos, os tengo que recordar un poco la parte importante que es el paciente. Y con eso, I'm going to turn that to you, Jude. You don't know what I have said, but...
Can you hear me okay? I always ask Javier, what did he say? And he said, don't worry about it. I was telling him we're going to order pizza. And that was it. I have a lot of people I need to thank. Beatrice for saving my life. Thank you. I have a lot of dear friends here. The first introduction I had to Spain was through Jesus and our collaboration that's been going on for years. I told a quick story. He came to visit me in North Carolina, and I had 2 young children at that time. And I was running to take one to swimming and one to this, and I didn't know what to do with Jesus, so I dropped him off at my mother's house. And I said, I'll be back in 3 minutes, I promise you. I raced to get my kids, came back, they were gone. Jesus was gone, my mom was gone. I'm like, oh my gosh, I lost the scientists. They decided they go to church together. So neither one of them ever miss Mass. They go to Mass every day. So I'm sitting there anxious. They drive up. Where were you? We went to church. I lost my friend to my mother. So today I really want to share with you, it's been a journey. It's taken too long. It's about 40-something years from when it started, and I'm going to try to walk you through the things we did that were problems, the things we solved, the things we haven't solved. And I got a new program on my computer, so it looks real fancy. This is our image of an AAV particle now. But I want to tell you a story that starts back in 1953 that goes to 2022. And I participated coming in here around 1970. And when people ask about gene therapy, when did it start, how did it start, I failed to realize that gene therapy was going on almost 200 years ago. This is an image of Jenner, who was vaccinating his own son with smallpox, a cowpox. He noticed the observation that the women that worked with cows never got smallpox when the epidemics came through. So he had a hypothesis that if you got vaccinated with the cowpox, it would protect you from smallpox. So he actually infected his own son with cowpox. What he did was a gene therapy experiment where he put genetic information outside of the body, inside the body, and it educated the immune system to protect him against smallpox. And so while we think about it being very sophisticated, it was done a long time ago. I went to China and I gave this presentation, and the Chinese were very polite, very humble, and one tapped me on the shoulder and he said, "We were doing smallpox vaccine in the 14th century." I was like, okay, I'm sorry. So gene therapy's been going on for many, many, many years. This is a magazine called the Saturday Evening Post, and it illustrates a young boy being in what's referred to as the iron lung that used to keep you alive if you had polio, because poliovirus would infect the nerves and would cause paralysis. And they give the image as if everybody's sitting at home playing around while Jimmy's in the iron lung. But the reality of it was that the wards looked more like this. Hundreds, if not thousands, of kids were being kept alive by the iron lung. And it was because of this epidemic that there was the March of Dimes trying to find a cure or a vaccine for polio. What you didn't appreciate was that Jonas Salk was the first to come up with a vaccine that was used for polio in 1953. But during these epidemics, people used to flee the city because they didn't know what caused the infection. They used to leave the city. You could not go into certain towns if you were below a certain age because they were scared that kids carried the viruses. You would end up finding out that children could not go into movie theaters, so there was such paranoia. There was such misinformation. The Italian immigrants did not get polio because of their social behavior exposure. They were gangs that used to beat the Italians up because they thought they caused the virus. Talk about misinformation. They actually thought cats caused polio virus, and they killed 70,000 animals trying to get rid of something that had nothing to do with cats. So you think about COVID today, and we fight over where to wear a mask or not to wear a mask. History repeats itself. 1953, because of that, there was an effort to try to find viruses to make vaccines. And the virus that they found was called adenovirus. It was taken out of the adenoid tissues. They thought it caused the common cold because people had upper respiratory infections. And so what they were doing was isolating adenovirus, trying to make a vaccine to prevent people from getting sick in the winter. And when you look at it under the electron microscope, every one of these is an adenovirus, but then there's this tiny little particle here. And that particle, when they purified it, put it on cells, it did nothing. So they thought it was a precursor protein that made the adenovirus, and they dismissed it. So for 10 years, it was not understood, but every time they grew adenovirus, this virus would show up. So they called it adeno-associated virus. It was always associated with this infection. And that's how the name came. But it was some biochemists unrelated to virology that came in and found out that it was a non-envelope virus, it was a single-stranded genome, and it was non-pathogenic. This is an experiment you can't do today. They took adenovirus and they went to an orphanage, and they gave kids infections with adenovirus that was free of AAV and adenovirus that had AAV, and they were looking at the severity of the disease, and they showed there was no difference. So they determined that this was a non-pathogenic virus that was like a molecular hitchhiker. So the life cycle eventually got figured out that it had a lytic component, which meant in the presence of adenovirus, AAV, both of these would replicate in the nucleus and produce progeny. But if you took AAV, like in that electron micrograph, and put it in the cell, it went latent. It didn't carry out any molecular steps. It stayed latent. If you've ever been infected with chicken pox, your virus is latent. You'll end up getting shingles, which is herpes zoster. It's the same virus that you get as a child, you carry for 60, 70 years. So this became a very interesting paradigm, because if we could control this part of the life cycle, we could use this as a delivery system to put genes back into cells. Okay. 1971, there was a National Cancer Act that was signed into law in the US. It was signed by Richard Nixon, and it was a commitment of $1 billion to study cancer, to figure out finally what caused cancer. At the moment, the only molecular tools that was available were viruses like herpes virus, adeno, SV40. They would cause tumors in animals, so there was a lot of research being put into understanding how did these viruses actually take over a cell and turn it into a tumor. And this is the little parvovirus. And so in 1971, the money started to flow into the field. This is 1978, and again, it's amazing how dumb you are when you're young. My job was to clone AAV. And I cloned it into a bacterial plasmid. The plasmid was called PBR322. No one understood what PBR stand for. It was done in one guy's lab, and he named it after Pabst Blue Ribbon beer. 322 bottles later, he came up with this plasmid. So there are all kind of names out there that people aren't aware of, but we cloned it into PBR322, and we drank a case of beer. And we were able to get the first infectious clone that if we physically put this into cells and then chased it with adenovirus, it would jump out of the plasmid, replicate, and produce progeny. So we could recapitulate the entire life cycle with a piece of nucleic acid. That same DNA, if you put it in cells and you didn't add adenovirus, was totally inert. So it had no function. So this allowed me to jump to Princeton. When I was at Princeton, I was able to remove the AAV genes, which are the replication genes, and the capsid genes that form the structural. And we were able to show that we could remove 96% of the sequences and put foreign DNA in between here. And then we could package it back into an AAV particle. And that was, oops, time flies. Sorry about that. Okay, we'll just go forward. Sorry. We ended up putting reporter genes in. This is an example of the luciferase gene put into the virus. You take the virus and stick it into the animal. You can see everywhere the virus is simply by the gene being expressed. Before that, we had to sacrifice the animal and look at a reaction like LAC-Z, which would turn cells blue. So every animal time point was sacrificed. Now it's all real-time imaging. This took me to my first job at UNC, and this is the experiment that was done by Zhao, who deserves all the credit for this, where he took an AAV vector and he put a gene in it, but what was important, he put it in the muscle of an immunocompetent mouse. Now realize, every other group, Richard Mulligan at MIT, Enda Vermeer at Salk, all of the powerhouse labs that were working on viruses were trying to deliver genes into animals. Every one of them was rejected by the immune system because it was seen as a foreign protein. But when we did this experiment, what Zhao was able to show was, one, if you increase the dose, you get more and more cells transduced. But more importantly, he came in the lab and said, Jude, it's positive. I said, it doesn't mean anything. The immune system's going to kill it. Jude, it's positive. Jude, it's positive. Jude is positive. It was 1.5 years later that we finally decided to publish this because we couldn't refute the fact that this was still being expressed in an animal 1.5 years. So it completely recapitulated the life cycle of AAV, that it would go into a cell and not cause death or any kind of damage. As a result of that, the concept of using AAV to put in multiple genes, and my concept of referring to it as a molecular FedEx truck, was that you now had a package that you could have put in here, and these would go through zip codes to take it to different target tissues. And this is what I want to share with you and how the field evolved. The first clinical trial to go into humans was related to cystic fibrosis. And if you think about it, it was I'm sorry, this is being difficult. Let's try it one more time. In 1996, Terry Flott, who was at the NIH, decided it would be the perfect virus for cystic fibrosis. The virus was an upper respiratory infection. The gene was a single gene defect. So all we needed to do was simply put the virus back into the lung, let it deliver the CF protein, and the channel would rescue the disease. And they did this experiment, but absolutely nothing happened. They got no results. Everybody was pulling their hair. Why didn't it work? In the animals, it looked positive. And the reason was we ended up that we didn't understand how the virus actually infected cells. We had very limited experience with vectors, and clinical trials are very, very hard. So the lacking of the biology, I went back to the lab and I focused my entire lab on trying to understand what were the infectious cycles required for AAV. We first showed that the primary receptor was heparin sulfate. We ended up showing that integrin is a co-receptor. We ended up following after that what were the infectious pathways, and what this led to was a body of information that began to explain how the virus would bind to the surface, go in through clathrin-coated pits, escape from the endosome, go to the nucleus, go in through a nuclear pore based on nuclear localization signals, and here it would release its DNA, and as a consequence of that, the gene would now turn into a double-stranded DNA and express the protein. What we found out was when we went back to human lungs and we looked, there was no heparin sulfate on the surface. It was perfectly explainable why AAV would never get into a lung cell. In fact, the heparin was between the tight junctions. And so you needed the cells to be torn apart before AAV would. And that's exactly what adenovirus did. It would infect a cell, kill it, tear it apart, and AAV would jump in for the ride. Man, I'm really messing up on this one. So after that, we ended up developing a mechanism where we could package any AAV serotype into an AAV helper. That let us put multiple vectors at work. So now you have a single transgene. You could basically dial in whatever capsid you want, and that capsid could be put into animals, and you could determine which one was superior for going into muscle, lung, heart, eye, so forth and so on. And this is what's emerged in the field at the moment, at picking the right virus to go into the right target cell. As a result of that, the first efforts were put into the eye and the brain, and I want to share with you what was done for Leber's congenital amaurosis, or LCA. It's a single gene defect. There are multiple genes that are responsible. The eye is an easy target to get access, and the clinical endpoints is lack of vision or having vision. Here's an example of a dog model that has the disease, and what you'll see is that the dog has been treated in this eye, so when the caregiver walks behind him, he can't detect him, and then when he comes within vision, he can now track him, demonstrating that he regained his vision. Is that me? I thought it was siesta time or something. Florian, get out of here. This is an example of a patient. Can you turn it on?
6 times and was disoriented twice. It took him 77 seconds to finish the course. 6 months after treatment, the patient showed a dramatic improvement. He was able to make his way through the maze in 14 seconds without bumping into anything or getting disoriented. He was one of 4 patients that showed an improvement. The treatment also proved to be safe.
So this is now the first drug in the U.S. to be approved for the eye. Since then, I wanted to share with you that the product was more than adequate production. AAV2 is okay if you're putting it directly into a target. And clinical trials are still hard because it took basically 10 years to get this through the studies. So what did they actually do in these experiments? They were doing what's called a subretinal injection. This is where they remove all the vitreal fluid, inject the virus underneath, so you actually detach the membrane and let it reform to get it in. And we were quite aware of the fact that if you use the technique that was used for monoclonal antibodies, this is a vitreal injection where they just numb the eye, inject the antibodies. It's an outpatient procedure. And like the person in the third row, I can't stand to look at this, but this is what thousands of people do every 2 weeks to get their therapy. And so we were looking for, can AAV be given vitreously so that you did not have to detach the retina? And we were able to determine that AAV2 could transduce through the vitreous and transduce the target cells. And what we were able to do was take the atomic structure and all of the other serotypes that were out there, and we could determine that it was actually the AAV's heparin sulfate binding motif that was responsible for causing the virus to get into the cells. This is showing you a single subunit of the AAV protein. I'm now going to put this into a pentamer, use this as your Lego to snap it together. And you can see it's snapped over its partner. Here's where the mutations are being generated. If you then put that on a virus, here's your 5-fold symmetry. And then there's 12 of these that form a particle. And so when you look at it, even though we changed 2 amino acids here, there's 60 changes across the entire surface. So it's been punctated with a mutation. And if you take that and put it in animals, you can see we made an AAV2 behave more like an AAV1 as far as going into muscle by stitching in these amino acids into that location. And we were able to take this location and we put all 20 amino acids into this one spot. And you can see by the loops how they slightly change that it caused it to fold or bend in different directions. And each one of these were evaluated for its ability to transduce. And what I'm showing you here is actually the receptor of AAV2. This is the positive charge, this is the negative charge. You see when we make the mutations, we start to lose various charges and get more of the other. Here's what happens when you look at it from an electrostatic potential. You get more charge differences. Here's what happens when you put it in an animal. This is AAV2, this is a point mutant, here's another point mutant. This is AAV8, which does not carry heparin. This is AAV8 after we put a heparin binding site on that capsid. So we could take a virus that never went into the eye and make it go somewhere by changing the zip code on the surface, by primarily interfering with the glycoproteins that are responsible for binding. This is a clinical trial that's going to start soon. It's called cornea clouding, where the enzyme's missing, and as a result, the protein builds up. These patients end up getting cornea transplants over and over and over. What we ended up doing was putting the virus in there. There's a dog model. When you shine the light, it's refractory when it's cloudy. When you put in the vector, you can tell which dog got the vector. These are 3 separate animals that have supported the clinical trial going forward in humans. Outpatient, 10 to the 9 viruses, 30-second procedure. This is an effort we started way back in 2002. It was primarily parents coming in asking for help. It's a disease called Canavan's disease. You're missing the ASPA gene, and this single gene is responsible for a pathway that's required. We ended up doing the clinical trial where we injected AAV2. We found out that the gene was indeed in the patients, and it's provided success. I'm so sorry. I wanted to show you this because this was our first clinical production we put in a patient. And you have to appreciate that this is a maybe Thermo Fisher protein marker here. This is the purified virus. And when we gave that to the FDA, we said, we can't get it any cleaner. They were very supportive, and they said, well, Jude, all of these proteins came out of a human cell, so you're just putting human protein back in. We'll let you go forward, which was amazing, but I think it was a real act, one of the first commitments to make this field go forward. As a result of that, we had to spend a number of years figuring out how to get rid of these contaminants. I don't know why this is so difficult. And we were successful, and this is the product that we finally got to. The 3 capsid proteins, VP1, 2, and 3, essentially removed everything else that was a contaminant. So we actually entered the world of pharma where you had to develop things to a level of reproducibility. Lessons learned from this in the cannabis disease was protocols that we had to establish. We actually found out a very simple thing that almost killed us, and that was if you took the virus and did the mouse experiments, it worked perfectly. If you took it, put it in the apparatus that the FDA said you could use on human, nothing happened. And the reason was the virus stuck to the tubing. We had never used the tubing that the FDA said was you had to use if you were using humans. So until we found that out by accident, we had to flood the tubing with AAV particles to saturate it, and then put the therapeutic virus in to treat the patient. And that's allowed our colleagues, Chris Bankowitz and others, to go after other diseases. This is called amino acid decarboxylase deficiency. It's also referred to as juvenile Parkinson. They're missing this single gene. This gene is responsible for converting L-dopa into dopamine. So Chris and them were injecting the virus into the substantia nigra using MRI-guided delivery. This is going into the patient's brain. And I hope the videos come up. This is baseline. If you see the patient, almost all movement is done by the caregiver or the parents. And basically, after a single injection at 18 months, you can see her basically dancing on the couch. And here she is at 24 months. She's actually having a conversation with her nurse. The nurse asked her if she wants to eat. I think it was broccoli. You'll see her response in a second. Like, do you want some broccoli? She looks at you like you're crazy. And then the next thing is, no way. No way. So this is a remarkable result that Chris has been able to carry out, and it's through the Children's Foundation in Columbus that actually Javier and Sheila and others started. They're treating all these patients for free. There's 191 of them. 32 have been treated. This may be the first disease that gets eradicated off the planet simply by making enough vector and getting access to these patients. I'll show you this real quick because this is a story that's going on and on. In 2006, hemophilia was the popular disease, single gene defect, liver's the target, give it IV, measured clinical endpoints by measuring protein. We demonstrated in the dog it would work. Kathy Hyde took it into patients, and whoa, she got therapy, and then a month later it disappeared. We had never seen this before in any animal model, so all of a sudden the whole field turned upside down. We couldn't explain what happened. And as a result of that, we began to realize that it was transient therapeutics in the liver. It was the first evidence of AAV toxicity, and that animal models were misleading. So the common nomenclature is mice lie and monkey never tell the truth. And if Trump gets in office, you get the repeat of everything. But what we did was we decided to work on the fact that if you stay below a certain level of vector, you could get past this toxicity. And so we developed what's called self-complementary vectors by removing the ability to replicate one end. So now when we replicated it, we would package a double-stranded DNA molecule into the AAV particle. This double-stranded molecule was more potent than the single strand, so it allowed you to use a much lower dose. And so here's an example at 10 to the 10th particles in the liver. You see a GFP. You see at 10 to the 12th particles a single strand. However, if you put in 10 to the 10th a double-stranded, you can see you get almost the same amount of transduction, 2 logs lower. So this became a very popular reagent. We developed this basic science. It went into 2 clinical trials, into the first hemophilia B trial, and then it went into SMA trial. The hemophilia data right now, these patients are out over 11 years carrying the self-complementary vectors and still expressing. And as a result of this, it became clear that if we could continue to make these improvements, we would get more drugs on the market. I'm sorry, this is the patient who got treated with the SMA drug. This is called floppy baby disease because they can't hold their body upper muscle. And so this was done in 2019. It was put in IV. It had to cross the blood-brain barrier, over 400 miles of delivery. And so as administration became an important point, actually Novartis was trying to start a new clinical trial by doing intrathecal so they could get less virus to the target without going through IV. Unfortunately, we started to see toxicity in children, so even adults were not predictive. This caused the FDA to have some specific meetings on what's the risk of AAV. And as a result, we all are becoming very familiar with these so-called issues with AAV, pre-existing antibodies, repeat dosing, immune response, large-scale production. And as a result of that, the field has shifted to try to go back and understand some of the AAV biology that's required to make this last step more efficient. And I'm going to share with you the 4 pillars that are now used is the capsid, the transgene, the promoter, and production. And I'm going to talk to you about a capsid just for 2 examples, and then I'll stop. This is an example where we developed a new AAV virus that would detarget the liver. So we're trying to get away from the toxicity that everybody was seeing, but we wanted it to go into muscle, so we wanted it to have muscle tropism. The way we did this was we took an AAV2 where we knew the heparin motif was. We took 7 amino acids from AAV8, we stitched them into AAV2, and we called it AAV2I8. And you could see while AAV2 went into the liver like AAV8, this one detargeted the liver and had a tropism for the heart. We were able to confirm this in monkeys, and we compared to AAV9, looking at different muscle groups, and you can see it's detargeted in the liver. And as a result of that, we started in a clinical trial for congestive heart failure, where we're putting the virus down the descending aorta, letting it infect the heart, and trying to put back a gene that would be responsible for providing better cardiac contraction. This is how patients are rated if you have congestive heart failure. They're called the New York classification, class 1, class 2, class 3. Class 4 means if you don't get a transplant, you're not going to make it. There's only 2,000 transplants a year, 500,000 patients. Class 3 is one where you're severely limited in what you can do. So Sheila was responsible for facilitating this trial with Raja Hajar. Our first patients were all class 3. It happened right during COVID. So they got the vector. We couldn't monitor them. We couldn't get them. So they were texting us, oh, I feel great, having a good time, blah. Like, placebo effect, placebo. We ended up getting our patient to come in, and she was reclassified by a totally different physician from a class 3 to a class 1. Our second patient was a 7'1" patient who said he couldn't hit golf balls because he was too exhausted. He was now hitting golf balls, walking to get them and bring them back. And this is now going into pivotal studies. Again, based on the fact that we developed a new capsid that had very specific features based on what we learned from the biology. This is actually a biopsy from the heart. We took out very specific tissues and we carried out PCR studies to see how many genomes were inside of the heart. We compared them to the pig study, which is what we had to use for the FDA. And we ended up determining that this new capsid was actually 2 logs higher in the human heart than it was in the pig heart, because it was designed to go into humans. So I think I'm trying to leave you with, the field is getting a little bit more sophisticated in designing delivery systems to go where you want them to go. This is a classic example of using AAV libraries, an example where epilepsy is the target. We're looking for a novel technique where we can take an IV injection and put it into the host and find a virus that will cross and get into the target tissue. This was done by taking AAV vectors, shuffling them, PCR them together, generating a library, and then selecting for a very specific capsid. The way this was done in the lab was we took a rat that had a seizure, injected the library IV, and then a day or so later, we would pull out tissue, PCR, whatever virus ended up in the brain. We clone it back into PBR322. We would make new virus, and this would cycle around 4 or 5 times to keep purging the system to look for a virus that would go specifically to the target cells. And here's an example. We ended up publishing this, that we got a virus that would cross the blood-brain barrier. And this was a study looking at the ability to put a gallon in. You'll see the rat here, when he gets an electrical impulse, he'll have a seizure, uncontrolled, which is really horrible. And this is the animal that's been treated. You'll see the electrical shock right there, and the seizure is completely blunted. So there's the potential to go in to these etiological diseases that don't respond to drugs with a genetic approach. I think I'm going to spend the last second talking to you about production, because we all know that everybody is very, very sophisticated in making new AAVs and new promoters and new transgenes. This is something that took us over 20 years to develop the Pro-10 cell line that we ended up mastering and transferring here to Spain, our colleagues at Virogen. And it is a Virogen template that Javier, Sheila, and a number of Basque officials were able to get us a footprint where we could actually start growing AAV in a very reproducible way at GMP levels. They now completed the production facility for a 30,000 meter facility, 300,000 square feet if you're American. And this was the drawing of the facility. This was us last September, Carmen, Sheila, I don't know who he is, and our Laura. And actually, the facility was coming online. I think we got 2 modules or 1 module. 1 module is online, the other 2 have been finished. In less than 5 years, we've gone from about 8 or 9 people to over 300 people that are now working there. This footprint's going to continue to grow because of the demand for AAV, and the demand for these genetic drugs is so high. So I think I'm going to stop by simply giving you this so-called journey. Started with trying to figure out how to put the same gene in different viruses. We ended up making self-complementary vectors to solve a toxicity problem. And it was because of the immune response, we designed vectors that would detarget the liver and go only to the heart. And we now are more excited about avoiding routes of administration, but letting viruses do the targeting. And this also is a critical component, which is gene regulation. You can't see the many, many people there, but I did it by myself. I just show this slide all the time. Thank you. That's it.
Thank you, Jude.
Questions?
Any questions?
Florian.
Thank you so much, Jude. I'm part of the Thermo Fisher crowd as well.
Discount.
I have a question regarding the origin of AAV. And yeah, in terms of the evolutionary origin, and if there are any other similar associated viruses, or you were looking for them.
There is an AAV for almost every species. There's a human AAV, there's a canine AAV, there's a feline, even down into crickets. What you see if you go into the parvovirus family, they form two groups. One's called the autonomous parvoviruses, and the other one's called the dependent parvovirus. The autonomous parvovirus, like canine parvovirus, causes disease in animals. B19 is the human equivalent. It causes infection of erythroid progenitor cells. It's referred to as slap cheek disease because the red cells complex underneath the skin. It looks like you've been slapped. If you're a sickle cell patient, you'll die from that infection because you don't make enough red blood cells anyway. Those viruses only work in rapidly dividing cells. So canine parvovirus works in the gut. The cells in the gut turn over every 3 days. So if the cell is going through the cell cycle, the virus knows how to propagate. The dependent-parvoviruses need a helper. And guess what the helper does? Adenovirus goes into a non-dividing cell, tells it to turn on, and it starts dividing, and AAV takes advantage of that division. So AAV has not been evolved to go into dividing cells because it can't find them. It goes with a virus that takes any cell and turns it into a replicating cell. So it's two different classes in which one is a symbiotic relationship between the helper virus and the AAV. As far as its role, it's a non-pathogenic virus. It doesn't seem to have any specific function. And there was an effort to start looking for molecular hitchhikers that may be in your body. But how do you do that? Do you do it if someone laughs? Typically, you look for viruses when you're sick, right? Fever, blotches. How do you look for something that doesn't cause disease? You dance a lot, or I don't know. So it's really hard to try to decide how to go pull out viruses, but I think more and more people think there are many, many molecular hitchhikers like AAV that just haven't been found. And as you can see, this was found purely by accident, looking for a vaccine to stop the common cold. So that's its history.
Thank you very much.
Oh, you're welcome.
If I may. Yeah, in terms of the tissues and this hunt for finding the AAVs that are going to be able to target different tissues, what are some of the most challenging ones? What are the ones that we're still missing and we are not able to target yet?
Actually, it's not what cells are the most difficult. It's how naive we are to what is involved in evolution. So everybody, including myself, we made our libraries. Our first experiment we had on a plastic dish, astrocytes, 100% astrocytes. We threw in the library, cycled it. We got a beautiful virus. It was called 1829. It was AAV1, AAV8, AAV2, and AAV9. Beautiful, 100% transduction in astrocytes. We took it, we stuck it in the mouse, 0. We couldn't figure out why, and then we realized astrocytes don't grow on plastic. And so we were selecting for a virus that grew on cells that never grow on plastic. So we had to go in vivo. And putting libraries in vivo, we thought we solved the problem. Gloria knows. A lot of us spent years chasing this. We get beautiful viruses. They work gorgeous in mice, but they don't work in monkeys. And then you say, oh, let's get smart. Let's select for them in monkeys and keep our fingers crossed that they're going to work in humans. And that's where the field is at the moment, simply making mistakes along the way. If we were fortunate like COVID, where you could take 300 million people and give them the virus and pull out the one that was best, we would have the best vector that you could ever imagine. But it's science, by going down rabbit holes and finding out all the wrong answers, if that makes sense.
Thank you. Thank you very much.
I have a question over here for $2. I tell you, that sun is killing me.
Thank you very much. I was amazed to see your the facilities built in Spain. How do you see? Congratulations. How do you foresee the future of this technology expanding into our healthcare system, and also how do you dream about it?
That's a really good question. So right now, if I were to look at this technology, I would say it's very similar to the very early days of cell phones. They were about the size of a shoebox. And when they developed it, I think they knew it was going to end up being a razor-thin cell phone, had more computing power than NASA. And so right now we have the shoebox. We're trying to make reagents while we're trying to improve the production. And it'll clearly go through steps of development where it won't take as many cells to produce as much virus. I think eventually we're going to have a synthetic system where you can do everything in a test tube, create the AAV, create the capsid, and package it. I think that's doable, but right now, that's probably an 8, 12, 15-year project. And the need for these therapeutics is so demanding that we can't walk away from what we're doing. So we're trying to create a reagent while we're trying to solve a problem. And it'll happen over time. I think it will absolutely happen. The next thing you're going to see is a transition from viruses to synthetic molecules, biological nanoparticles, where you may take a trafficking component of a virus and put it in a lipid nanoparticle and get something that's more dynamic than the virus itself. That's probably going to happen, and you'll see a transition where AAV will be more like the Model T car than it will be the Stingray. Let's put it that way. Question. You guys, you make me work for my dinner, I can tell you that.
Well, very thanks for the speech. It's from a scientist that left the science, the bench, and now working in Thermo Fisher. I just wonder how our company can support, in which way can support.
Discount. Oh.
The synthetic one, and the other question is how the synthetic, so how you working right now for the synthetic activity, or
so
is
let me take your question and start from the reality. Right now we have 10 to the 6 cells per mil. We know if we go to 10 to the 8th, we get more virus. We go higher than that, we get more virus. It's coming up with medias that let us stay in a small vessel, but a higher density. Very traditional problem that can be approached and tackled by the tons of experience at Thermo Fisher. What's the best media? What's the things that get exhausted quicker, glucose, CO2, whatever. That's probably a marriage that should be made in order to have the field go quicker. Right now, the new facility has a 2,000-liter bioreactor in it. The 2,000-liter for muscular dystrophy may treat 1,000 patients. There's 40,000 patients out there. So try to imagine how much you have to make. If you could take that same bioreactor and get 40,000 doses out of it, that's another disease that's off the list. So that can be done readily, right, in my opinion, by a concentrated effort trying to figure out the densities, the optimum expression. I apologize for those who aren't in this space, but if you took a CRISPR approach and knocked out every gene in the producer cell, you could find out immediately which ones are responsible for assembly, replication. You could turbocharge that cell to be more active than it is by just doing a knockout approach and finding the genes. The synthetic is going down a slightly different path. Right now, Javier and his colleagues have generated what we call the doggy bone DNA, where we synthetically make the AAV in a test tube, and it looks exactly like the virus. So you have one substrate. The next question is, can you get the structural proteins to come together and pull that substrate in. That's probably going to take some biochemistry where you start to find out which enzymes are required to thread the DNA inside of the capsid. That can be done, but that's going to be very FDA non-compliant, if you know what I'm saying. When they ask what's in there, you're going to say, I don't know. I'm just putting in an extract and it works. But eventually you'll pull out every protein and everything, purify them like we do, and you'll be able to do a synthetic. I give that about 7 or 10 years, depending on who's working on it. And I'm retiring, so don't look at me.
Thanks.
Are we done? Thank you.
Thank you, Jude.
Thank you.